Hey guys, all right, so today we're going to be talking about chapter 14, which is Speciation and Extinction. I did get a new haircut, and it's like a new expectant mother haircut. And, I feel like I'm almost there. I feel like I'm almost to full mom status. I just need like a LaCroix here with me, and then I'll be ready. But we're going to be talking about speciation and extinction today. So that's how new species come about via macroevolution and how species go extinct, okay, through many methods. So I'm going to share my screen and we're going to get going. Okay, so chapter 14. We're going to talk about what is a species first, how this sort of line of thought has changed over time. So in the 1700s, Carolus Linnaeus was the first to define sort of what he thought a species was, and he characterized it by appearance because that's basically all they had in the 1700s, right? They couldn't sequence the DNA. They didn't even know what DNA was yet. So he is quoted as saying, all examples of creatures that were alike in minute detail of body structure. So anything that was almost indistinguishable from another organism was going to be lumped together in a species. Now, this we know is problematic for a couple of different reasons. First of all, you can have organisms of the same species that do not look anything like each other. So for instance, males and females in a species, a lot of times will look quite different. I mean, think of some birds birds is a perfect example. So like female versus male cardinals or female peacocks versus male peacocks. They look literally completely different. So this is an issue. So he basically started this Linnaean classification system, which we will talk about more at the very end of this chapter. And it basically is just a hierarchical system for how to classify species. So he knew, and lots of people back then knew, you have these species that they thought were just organisms that most looked like one another, and then those organisms would also be related to other organisms and things like that. So this hierarchical system said you have a species, and then you have many species in a single genus, and then many genera within a family, and so on and so on and so on, getting this hierarchical system. So what I want you to write down, and we will talk about this, we'll come back to this later, but dear King Philip came over for good spaghetti. This is domain, kingdom, phylum, class, order, family, genus, species. So one more time, domain, kingdom, phylum, class, order, family, genus, species. Okay, that is the hierarchical classification system. So in a domain, you have millions of species. In a species, you only have one species, right? In a genus, you might have one species, like the genus Homo for Homo sapiens, or you might have thousands of species, like in the grasses in Poaceae. So actually Poaceae is a family, but you get my point. It's this hierarchical sort of system where you get more and more species in each one of these groupings as you go on. So each species name combines the broader classification of genus with the term species. That's what we're going to use most often. We're not really going to be talking much about domains, kingdoms, phylums, orders, and things like that in this class. We're mostly going to be referring to species and the genus that they're in. So Homo sapien is our genus, and species. Homo is the genus, sapien is the species, okay? And now the genus Homo is an interesting genus because at one point there were plenty of different species, many different species within the genus Homo. But now there's not. We only have one species in that genus, and it's Homo sapiens, which is us. So more recently, We have what's known as the biological species concept, which is going to do the same sort of thing as the Linnaean classification system, where they based it off of how an organism looks, except instead of going with how organisms look, we're basing what is a species on their potential to interbreed, and whether when those organisms interbreed and produce offspring, are their offspring fertile, okay? Because we have a ton of different examples of different species that are able to reproduce, but they do not produce fertile offspring. So they're from two different species. So just being physically able to reproduce does not make you one species. You have to be able to first interbreed and second produce fertile offspring. And again, it does not rely on physical appearances. Speciation is the formation of new species, and when speciation occurs, so let's say that just chop the world right in half. You have North and South America, and then you have Asia and Africa and Australia, okay? We're not including, you can include Antarctica, I don't care. Splitting it in half, and you have people from North and South America who are not able to migrate to the other half of the world, okay? To Asia, to Europe, to Africa, to Australia. There is no potential to interbreed because we physically cannot get over there. Well, then what's gonna happen is that these gene pools are gonna go down their own evolutionary paths based on their own environmental pressures, right? And then speciation will eventually happen. So it's when individuals of a species can no longer interbreed, whether it's because of a genetic mutation that means that they can't interbreed with each other anymore, whether it's because of a physical, like a physical block. So for instance, a mountainscape that doesn't allow them to cross over the mountain to interbreed with the rest of that population. Speciation, if a population is not able to reach another population, they will proceed down their own evolutionary lines and make two completely separate species. And this is a form of macroevolution where you have that speciation. So how can it occur? The gene pool of the population can be split by lots of different ways, and we're going to talk about them. One example is like a physical barrier. So this leads to microevolutionary changes where you have genetic drift, mutations, and natural selection in both of those populations that are physically separated from each other. Then after enough time, the population will have completely different gene pools from just moving down their own evolutionary paths, unable to reproduce together, unable to produce viable offspring. And now microevolution has actually become macroevolution. You've gone from these small changes in their gene pools of the population to macroevolution, because now not only do they have differences in their gene pools, they're also not able to interbreed, or if they are able to interbreed, they're not able to produce fertile offspring. Macroevolution, you had one species. Microevolution over many, many, many generations, now you've got two species. So note, determining if two similar looking organisms, if they are actually two different species, is nearly impossible, especially when you do not take into account DNA. So if you only are looking at organisms, it's almost impossible to tell whether or not they're different species, especially when you're looking at plants, for instance, like grasses, okay? It is almost impossible. Just because they look similar does not mean that they are the same species. And that makes sense, right? You have a ton of different bird species or fish or snakes that mimic each other, plants that mimic each other to get a selective advantage in their environment. So just because they look alike does not mean that they're the same species. If they can produce fertile offspring is a much less objective observation. So if you were in a situation where you had no access to DNA technology, you could not determine genetically how similar these organisms were, We only were able to either look at them or observe how they're actually able to breed together, observing how they are able to reproduce is a much more objective sort of measure of whether these two organisms are the same species. So this one up here, this is like the Linnaean classification system. Down here, this is like the biological species concept. All right, now guys, the biological species concept, it is better than the Linnaean classification system, where we're classifying them based on these minute details of their structures, but it still raises some questions. It's better, but it does not apply to all organisms, to all living organisms. So first of all, and these are going to be important for your exam and your quizzes coming up. So it cannot apply to asexually reproducing organisms. That's a big one, guys, because we have a ton of asexually reproducing organisms. Now, why? Think about this. Why? I would ask you this question if we were in class together, but I just can't gauge your reaction right now. Why can the biological species concept not apply to asexually reproducing organisms? And I want you to think about that. It's also impossible to apply to extinct organisms. Extinct organisms that we know of, that we know lived at one point from fossils, for instance. Why is it impossible to use the biological species concept and apply it to extinct organisms? Why? Some species have the potential to interbreed in captivity. This is such an important one. Okay, so I keep bringing up like how similar grasses are and things. When I was in grad school, I studied grasses, and that sounds so super interesting. And it like wasn't. No, it was. But when I was in grad school, there are these seed vaults all over the world, and they are incredible. Okay. And, just harnessing genetic variation in populations of species all over the world, and it's incredible. So in grad school, I was studying genetic differences and evolutionary relationships between grasses, and I was ordering these seeds from everywhere. And you could get a species from the United States, let's say it's from Utah, to breed with a completely different species, like from Zimbabwe, okay? They would be able to breed with each other and make a hybrid. Would they ever actually do that? No. In like real ass world, would the species from Utah ever make its way over to Zimbabwe, plant itself, and be able to just like ravage the world with all of its little grass babies? Like, no, it wouldn't. So because some species have the potential to interbreed in captivity, well, that's difficult because biologically, would they if it wasn't for us? Another example of that would be like ligers. Is a liger a thing like in the wild? Do they just go around messing around in the wild? No, not at all. You have two completely separate species and they make a liger. Okay, does that mean they're the same species? No, not at all. Just because they have the potential to interbreed does not mean that they are the same species, because remember, you have to be able to interbreed, and then you have to be able to produce fertile offspring, meaning that those offspring have to also be able to reproduce. And then number 4, for some species, reproductive isolation is not absolute. So this is just like my example of grasses, okay? See, I have plants right here. So for some species, reproductive isolation is not absolute, meaning that their gene pools are completely separated. They have no opportunity to reproduce, but they still could. Okay, so these are similar. All right, biological species concept, it doesn't have a perfect way to determine these boundaries of each species. So for instance, why are these separate species able to physically reproduce and create a new offspring, create a viable offspring, because there are different levels of this, right? You can try to reproduce and not make an offspring. You could reproduce and make a viable offspring, meaning that that offspring lives as a hybrid, and then you can make an offspring that is viable and also is able to reproduce. Okay, so there are different levels of that, and there's no real perfect way to parse apart these boundaries. So to fill in these gaps and to determine how closely related species are to each other, we use DNA sequencing all the time. We use DNA sequencing. If two organisms are more than 97% identical, then they are considered the same species. More than 97% identical, then they are considered the same species. Now, what's important is that more than 97% identical is talking about their shared genes, okay? Only looking at the genes that they share. So for instance, if we're talking about us versus chimps, there are genes that we have that chimps do not, okay? So you wouldn't include that. There's no way to tell how similar those genes are because the chimp does not have them. The same thing with us versus a mouse. There are genes that mice have that we do not. So we wouldn't include those. We're looking at, okay, between us and a chimp and a mouse, we're all mammals. We're all mammals. We all have mitochondria. We all have these things that there are specific genes for, okay? That every mammal or everything with mitochondria would have, okay? So we can compare the shared genes, and if those shared genes are 97% identical, you're the same species, okay? Now, between, I was looking this up right beforehand, between us and chimps, our shared genes are 96% identical, 96% identical. And then between us and mice, it's about 85% identical with those shared genes, okay? So us and chimps, we are not the same species, but we're very closely related to them. They have almost everything that we have. It's just a fact. It's just a fact. And then between us and mice, also incredibly similar, but like, do we look just like either of these species? No. Could Carolus linnaeus even predicted that we were as closely related to chimps as we actually are? No, probably not. So despite the challenges, reproductive isolation remains the most common criterion used to define a species, and we will discuss reproductive isolation in just a little bit. Okay, here is a clicker question that we would have had in class. Biological species concept cannot be applied to which of these? Can the biological species concept apply to humans? Yes. Can the biological species concept apply to plants? Yes, of course. Can the biological species concept apply to extinct organisms? No, it cannot. And remember, I asked you to think about why. And then, can the biological species concept be applied to organisms that look alike? Of course. We're just going to look at whether or not they can interbreed and produce fertile offspring. That's going to be our criteria for them. Another one, how does the biological species concept differ from Linnaeus's definition of the term species? Okay, so let's go through these. Linnaeus's definition includes how species look and their ability to reproduce. Linnaeus's definition only included the ability to reproduce. Okay, first of all, are either of these true? No, they're not true. Did Carolus Linnaeus take into account the ability to reproduce in determining what a species was? No. If you'll remember, he said all organisms that were alike in minute detail of their structures. Okay, so how they looked, exclusively how they looked. So A and B are not it. The biological species concept includes species anatomy and ability to reproduce. And D, the biological species concept does not classify species by anatomical similarities. So the correct answer is D. Yeah, because, all right, includes species anatomy and the ability to reproduce. It doesn't matter with the biological species concept, if organisms look the same, if they can reproduce and produce fertile offspring, they're the same species. So for instance, like that, the male and female peacocks, for instance. Okay. So D is your correct answer. All right, reproductive barriers. If the potential to interbreed defines species as we know it through the biological species concept, then reproductive isolation will result in new species. And that's because as you separate physically or genetically, if you have any sort of reproductive isolation, and we're going to go through all the different ways that you can reproductively isolate a species, they have totally separate gene pools now. They're not able to reproduce, and they're going to proceed down their own evolutionary paths, microevolution leading to macroevolution. My husband's right there trying to take a nap. So don't judge. All right, guys. So we're going to start off with these 5 different examples of what are known as pre-zygotic reproductive barriers. So a pre-zygotic, let's go over this. Pre means before. Zygotic is referring to the zygote. Remember, when a sperm and egg come together, they make a single-celled zygote. And that zygote is going to divide via mitosis, make millions of cells, make an embryo, and then make a whole new thing. Hold on. You cool? Okay, pre-zygotic, so before you have a zygote, okay, before you can even make a zygote. So it's going to basically prevent a zygote from forming. So pre-zygotic reproductive barriers prevent fertilization. You will not get a zygote with pre-zygotic reproductive barriers. So your first barrier is called habitat isolation. So when you have different habitats, that is a way of separating these organisms, okay? So habitat isolation, you've got different environments. These are ladybugs that feed on different plants. If they feed on different plants, you've got different selective pressures, okay? They're gonna proceed down their own evolutionary paths. Microevolution leads to macroevolution. Now you've got 2 different species. Temporal isolation, when they are active or fertile at different times, like these field crickets, okay? So if they are fertile, for instance, in the summer versus in the fall, I guess that this would be the fall, would this species be able to mate with this species? No, they wouldn't, okay? Because they're active or fertile at different times. Behavioral isolation, if you have different courtship activities, you won't attract the females of this other species, right? So if this frog has this particular mating call, okay, this frequency of this mating call, and this other frog has this totally different mating call, is this frog going to attract the same mates as this frog? No. So you have isolation, you have reproductive isolation, microevolution leads to macroevolution. Now you've got 2 completely separate species. Mechanical evolution, this is when mating organs or pollinators are incompatible. So for instance, if you have these 2 different sage species and they have different pollinators, different selective pressures, microevolution to macroevolution, 2 separate species. Then you have gametic isolation. If your gametes, your sperm or your egg cells cannot unite, it's going to prevent fertilization. You're not going to be able to fertilize them. You're not going to create an offspring. Okay, and you can see that right here. All right, now we have post-zygotic reproductive barriers. So remember, before, all of those were preventing you from forming a zygote, preventing fertilization, whereas your post-zygotic reproductive barriers, post is after, zygotic is referring to the zygote. So you can produce a zygote, but we've got different reproductive barriers that can happen after you form the zygote. Okay, so reduce the fitness of offspring produced by members of two different species. So first, you've got hybrid inviability, where what you'll have is you create a hybrid, but for instance, the seeds or the seedlings are not viable, meaning that they won't, even if they produce seeds, those seeds are not, they're not going to work. Okay, so hybrid inviability. Here you can see those seed pods not going to be able to turn into this eucalyptus tree. Next, you have hybrid infertility or sterility. Hybrid infertility or hybrid sterility is just like this liger example. You have two separate species, the lion and the tiger, that are making this liger hybrid, and this liger hybrid is not able to reproduce itself. It's infertile. That's just like horses mating with donkeys. Horses mating with donkeys make mules, which a mule is a hybrid. Is the mule able to live? Yes. Is the mule able to reproduce? No. Okay. So hybrid infertility, where you make an offspring, but that offspring just isn't able to reproduce itself. Then you have hybrid breakdown, where, okay, hybrid breakdown is interesting because you have offspring, those offspring are able to reproduce, but when those hybrids reproduce, their offspring, or even the offspring after that, they're going to be more and more malformed, like just a breakdown of being able to be functional, okay, in different generations. So offspring of hybrid mosquitoes, so you had a hybrid, those hybrids did reproduce, and they have abnormal genitalia. That is hybrid breakdown. All right, so those are all of your different examples. Post-zygotic reproductive barriers prevent the development of fertile offspring. You can make a hybrid, and that's fine. Making a hybrid offspring that lives is not the point of the biological species concept. It's making a hybrid that lives and can generation after generation after generation reproduce and be completely fine, okay? Which of the following is an example of a post-zygotic reproductive barrier? Which of the following is an example of a post-zygotic reproductive barrier? So post-zygotic, meaning that it's after the zygote, you have formed an offspring already, okay? A cross between a horse and a donkey produces an infertile mule. Two types of fireflies lure mates with different flash patterns. Bees pollinate one mimulus plant species, hummingbirds pollinate another. Pine tree sperm cannot fertilize spruce tree eggs. Okay, so all of these three, B, C, and D, these are all pre-zygotic reproductive barriers, meaning that because you have the different flash patterns, because you have different pollinators, because you have different sperm and eggs that are not going to come together, gametic isolation, you will not be able to produce a zygote in these three examples. Versus cross between a horse and a donkey produces an infertile mule. You're producing an offspring. That offspring just has hybrid infertility. Okay. All right, guys. So your spatial patterns are going to define two types of speciation. What are you doing? No, what have you been doing?
Are you recording?
No, I paused it.
I kept getting much hair in my face and mouth, so I used the little brush to get off the hair of this kid and then laying on the couch.
I was like not gonna be surprised if you went and got the vacuum. It's like, what the fuck? Okay. No, but it's distracting. Lay down. Oh, babe. Okay, practice what you preach.
Babe, I'm fine.
Okay, lay down. What? What? All right, stop talking. These reproductive barriers are going to arise in two ways. You can have allopatric speciation, where you have a physical, like, separation of the species, and then you have sympatric speciation, where the species are physically together. They are physically there. They're able to, you know, they have the same sort of gene pool, but they're not able to make the conversion. They're not able to reproduce with one another. So kind of like, for instance, with the sage plants in the prezygotic reproductive barriers, how if they had different pollinators, then they were reproductively isolated. That's an example of sympatric speciation. All right, let's look at these. So allopatric speciation, you have a physical barrier that separates a population into two groups that cannot interbreed. So if you have them on two sides of a mountain, for instance, first of all, entirely different environmental pressures, okay? Entirely different. You have different wind patterns. You have different light patterns. You have different rain patterns, okay? So they're entirely different environmental selective pressures on these populations. So they will diverge, microevolution leads to macroevolution, and you've got two separate species. With no gene transfer between these populations, each one proceeds down its own evolutionary line. Eventually, these genetic differences will give rise to one or more reproductive barriers, meaning that even if they could come back together, like in a lab, for instance, or say that you had a, I don't know, say that you had a pollinator going over this whole mountain and pollinated one with the pollen from the other plant. After a while, no matter what, you would still have something like hybrid infertility or inviability or breakdown, okay? Because they're just too genetically different after a while of being physically separated. An example, a really good example of allopatric speciation are on the Galapagos Islands. So you have all of these islands, and on each of these islands, you have these Galapagos tortoises. And so they've diverged into several subspecies on the different islands because they're physically not able to get to each other to reproduce. Different evolutionary pressures, microevolution leads to macroevolution, leads to eventually different species. So they are in several different subspecies on different islands because, okay, subspecies, obviously, this is a man-made sort of term. And a lot of times what it'll mean is that these are species, like it is the same species, they could reproduce, they could make viable offspring, but they will not, because they are physically separated from each other. Meaning eventually, they're subspecies now, eventually they will be separate species. So that's allopatric speciation. Sympatric speciation, you have this continuous contact, they share a habitat, but they're going to diverge genetically. An example of sympatric speciation are cichlid fish. Okay, you have cichlid fish diversified into several species in an African lake. So if you're looking at this African lake, you're like, okay, like they're fish in a lake. Why are they different species? How could they have diversified? In a lake, you have many different habitats. Even if it's a small lake, even if it's a pond, you have different environments, different habitats in that lake, okay? So for example, you have one of these species of fish that are only hanging out in the shallow regions of this lake, and one that is hanging out in the very middle in this deeper part of the lake. Will they, if you have this fish living, mating, raising its young, eating, doing all of its stuff in the shallow regions of this lake, will it come into contact with the fish that are, hold on. Oh my gosh. Will they come into contact with each other? No, so that's automatically prezygotic reproductive barriers. They're physically in the same space, but are going to diverge genetically. That's sympatric speciation. Again, if they have a unique microenvironment, you've got reproductive isolation. Microevolution leads to macroevolution, leads to speciation. All right, another clicker question that you would have had. So let's do this. About 3 million years ago, the Isthmus of Panama closed, forming a land bridge connecting North and South America. Snapping shrimp collected from water on one side of the isthmus looked similar to those on the other side, but they cannot interbreed. What has occurred? Okay, so they look similar on both sides, but they cannot interbreed. Allopatric or sympatric speciation? Well, it's allopatric speciation. I want you to ask yourself why. I'm going to actually tell you. So because there's this land bridge that has physically separated them, that is literally just the definition of allopatric speciation. If these organisms are not able to physically interact and they proceed down their own evolutionary trajectory like alone, they are going to diverge genetically because of this separation. That's allopatric speciation. Now my nose is all itchy. Sorry, guys. So I want you to remember reproductive isolation and spatial isolation are important to speciation. So for instance, if you can't physically reproduce, you won't. If you have kids, have offspring that can't reproduce, you're done. If you can't reach something you could possibly mate successfully with, you won't, right? Other than the breeding and captivity examples that we were talking about earlier. All right, guys, I'm going to stop here and I'm going to pick up with the second part of chapter 14, which will be a little bit shorter.